Okay, no problem.
So yeah, I'll be here really early tomorrow to start those 5 plates.
Okay.
Yeah, I'll be here for a while tomorrow.
Sounds good.
Dinner.
Okay, see you. So this is a small kind of break for you guys. So we have a link going. We have.
When I send you the stuff for the competition thing, do you want the video of me presenting it too,
yes.
or no? Oh my god. Okay. You're gonna laugh.
Now I want. Okay, so we have a recording going.
And slides up, and I'll start admitting people.
Yeah, once we start doing this actual live thing, it kind of became less. It's just way too easy to do this way. Okay, if you guys need the link to the live stream, it's here. Where's the chat? Okay. All right, so that's the link. All right. I would suggest that whoever we're recording this anyway. Let's just start because we have to get on with our lives. All right. Okay, Jane, the floor is yours. Please start the meeting. All right.
Sorry, I think Jen slipped off for a second.
Okay, that's what happened. That's what happened. All right, Jen, we have you back. Yes. I hope. All right. I guess Gene is getting his tech together. So I'm going to take over for 1 microsecond. So thank you very much, everybody, to join our meeting. Today, we have a second day of the conference, Frontiers Innovation 2021. I'm going to hand this in 5 seconds to our moderators. We have 2 of them. We have a Gene Zong, who is our master person, and we have a Tyler, who is just besides him. Gene, the floor is yours.
Okay. Good morning, everyone. So let me get my screen set up properly before I got disconnected for a moment. Just 1 second here. I assume you guys can hear me, okay? So that's the first thing.
Yes, we can hear you perfectly fine.
Perfect, okay. So let me get a screen set up in the right order.
Beauty of technology, right?
I have been teaching this quarter, so I'm getting a little bit rusty on this. Yeah. Okay, it's almost ready. Just 1 second. Taking a little moment.
Well, it looks like most of the people believe that the pandemic will be here forever. It's kind of scary.
Well, that's the whole point behind it becoming an endemic, right? It's just part of the natural routine set of diseases that you get every season, sort of thing.
Yeah, we gotta live with it.
Well, but then it's technically not a pandemic anymore.
Yeah, that is true. Like flu.
I'm almost there, okay? Just 1 second. The screen's a little bit slow for some reason. My end. You know, I'm just gonna forget about the script. So I'm just gonna go by my freelancing. Yeah, so good morning or afternoon, depending on where you are. My name is Jin Jian. I'm very excited about this conference we are all in. I'm a professor in chemistry and biochemistry at UC Santa Cruz. I work on nanomaterials with little flavor of biotechnology. So mostly we focus on nano. So I'm very excited personally to learn more about talks from Professor Kim and Dr. Roscova about exciting topics at the nano-bio interface. And so we'll introduce them later with more details. But before we do that, I will give the stage or screen to Dr. Landansky and also Professor Manu Camps, so they can introduce the conference, and then we'll introduce the speakers.
Outstanding. Thank you very much, Dr. Zhang. So thank you very much, everybody, for joining us today. We have a joint conference courtesy of Dr. Camps and SHCI. We're doing this together because we believe that the ethics is important in the innovation, and it's one of the potential barriers to the innovation. Today, one of our topics that we're talking about at the conference is the nanotechnology. So everybody heard about the nanotechnology, but this is like one of the things that it's out there, and there are a lot of experts, and there's a lot of journalists, and there's nobody in between. And obviously, there's a high level of the anxiety and a lot of high level of the myth. So one of the things that we hope that we can do during those meetings is sort of talk a little bit, identify the barriers, and find a potential solution while we educate each other, having wonderful speakers about nanotechnology. We have two moderators. Again, Dr. Zhang is a little bit too modest. He has received the Bachelor of Degree in the Chemistry from the Fudan University in Shanghai, wonderful city. Been there a couple of times, and then he got a PhD in the physical chemistry at the University of Washington in Seattle. He joined the faculty of the University of California in Santa Cruz, where he's currently working as a full professor. Obviously, he does a lot of teaching. And he's one of those person who received in 2014 Richard E. Glenn Award from the ACS Energy and Fuel Division. The second moderator is Tyler. Tyler is one of those person who has a true PhD, quote unquote. He has a doctorate in the philosophy, not like I, who has a immunology. And every time he starts talking, the things rapidly became out of the hands because I don't understand what he says. He's so much ahead of curve. He works in the different function in the IEEE, where he works on the bunch of the standards and pretty much moves the needle a little bit further, especially when we're talking about establishing some kind of common ground for all of us. He was also very instrumental in creating this meeting, and honestly, this meeting would not happen without him. So now I'm going to hand this to Dr. Camps to introduce both the University of Santa Cruz and all the wonderful institutions they have over there, and maybe touch a little bit base about the SHCI.
Thank you, Dr. Laudansky. So so this is a collaboration between SHCI and the University of California in Santa Cruz. So SHCI is a society who really tries to figure out what are the technologies that are going to impact healthcare moving forward. So they're trying to stay like a step ahead and think about the ethical implications so that when we actually implement these technologies, we're prepared. So Crown College at UCSC is one of the 10 colleges, and our theme is the impact of technology on society. So our focus is more broad. It's not just focused on healthcare, but this year we've established this collaboration which really overlaps between the two institutions. We have two centers that are relevant. One is called the Center for Applied Ethics and Values of Advanced Technologies, and the other one is an organization called Get Virtual. I hope you enjoy this meeting. Thank you.
Okay.
Thank you
So,
both very
Jim,
much.
now it's up to you about the speakers.
Do you want to go over some of the things that we should pay attention during the talk first?
Absolutely. Give me 1 second. Thank you for reminding me that. It's right here. So we have a format.
So hopefully everybody can see this. So we have presentations followed by questions and answers and the discussion. And we may have a break up room for further discussion. So that's the schedule. Mm-hmm. And we also want to remind everyone of a few things to mute your audio during the presentation and position your camera properly, minimize visual distractions if possible. And so I think everybody can see it here. So we just want to remind everybody to be mindful through this remote venue. And so the conference can go smoothly. Thank you so much for everyone's attention.
Okay, so now I guess we start from the first presentation by Elena.
Well, shouldn't we introduce him first?
Yes, I think.
I think Jim just introduced them at the beginning, didn't he?
I think
No,
Tyler
no,
wants
no.
to maybe
He
say a
he
little
went a
more.
little bit into himself.
Yeah, go ahead.
Okay. And then there is a housekeeper right there.
Okay, so let me go ahead introduce Dr. Rozgova. She is a distinguished lecturer for the IEEE Nanotechnology Council. She has her PhD in chemistry from Moscow State Institute for Fine Chemical Technology. Also served a postdoc in Japan for JSPS at Tohoku University, and she's currently engaged in research at the Argonne National Laboratory in Chicago. And her focus is more on the general themes of nanobio interfaces, and she's been very engaged in these sorts of issues that we see in emerging nano applications in medicine. So go ahead and turn the time over to her.
Okay, so I'm going to turn off my sharing. Now it's your time. Go ahead.
So hello everyone. So let's say start slideshow. So do you see that? Can you see everything? And let's say start pointer, laser pointer. Actually, thank you very much. I'm very pleased to be here today. And I put a very general title to my presentation, nanoparticles from discovery to clinic. I should emphasize, since I work at Argonne National Laboratory, and we are mostly focusing on discovery part and letting medical doctors to continue toward clinical applications. And here also I like to show an image. This is a synchrotron image of single cancer cell with tiny micelle, polymeric micelle filled with iron oxide nanoparticles for delivery of cisplatinum to directly to nucleus of cancer cell. This is a very important image, and actually it's possible to use obtain it only using very modern synchrotron imaging. So also I need to tell I'm very honored to be part of Center for Nanoscale Materials at Argonne National Lab. It's a premier user facility providing expertise in cutting-edge instrumentation for whatever you want to do in nanotechnology and nano research, starting from design and analysis and synthesis and fabrication and application of nano. US Department of Energy funds 1, 2, 3, 4 nano centers, and we all work based on user proposals. And for users, after peer review, if this proposal is good enough, then use of all facility is free of charge. So whoever is interested to continue or just initiate work in nanotechnology, please visit us. And my group is nanophotonics and biofunctional structures. So since I'm a first speaker today, so I allow myself to introduce why nano and why nano, what is very special about nano. So US Nanotechnology, National Nanotechnology Initiative defines nanotechnology as understanding and control of matter at dimensions below 100 nanometers, where unique phenomena can result in new properties and new applications. So myself, since I tend to work in field of more green technologies, green chemistry, green nanotechnology, for me it's very important that scale of nanoscale is exactly the scale where much of biology happens. So examples, here I put some example. This is a ferritin, it's natural nanocage for storage of iron in bodies. Also, during some diseases, our tissues tend to serve as a template for biomineralization. For example, it's a bad process, iron oxide deposition during Alzheimer's disease. Also, I can give an example then. Magnetic particles are used by magnetotactic bacteria for movement, or some other microorganisms for energy transfer and synthesis of nanoscale nanoparticles. So second, why it's something special about nanotechnology is it makes possible directed evolution of properties. So I don't know how big this image is, but in very often, not only in bio-nano area, but in nano and technologies in general, we start from biological inspiration, like sunlight absorption and energy conversion. We utilize bottom-up and top-down technologies. Bottom-up is a synthetic, like synthesis, also synthetic biology, and top-down, it's microfabrication, and also we can say it's traditional biochemistry, and also an important part of self-assembly. Then these all particles, all materials can get together to more complex structures toward new or directed evolution of new materials and new property. So what's I can say there are four key moments about nanotechnology within today's presentation. Then size of materials or particles going down to nanoscale. Properties can change significantly comparing to properties at large scale. This is important. Second, playing with sizes at nanoscale allow for tunability of properties. For example, colors of gold nanoparticles depends on size of nanoparticles. Same about magnetic properties of nanoparticles can be tuned by size and sometimes it's very important shape of nanoparticle. So in nano, it's possible to make hybrids, to combining multiple functions in one hybrid structure. And also it's important if you compare with like medicines, for example, nanoparticles provide high surface area to volume ratio. All of this allows for multiple function, tuned specificity, and can result and can bring eventually to personalized medicines. So, but always, if you look at the third green square, so new technology is always connected, bring new questions and new concerns. So as for new technologies, nowadays, over 50 nanomedicines are FDA approved and over 100 in clinical trials. I think it's very modest numbers, maybe even more. And one of brightest example nowadays is Pfizer, BioNTech, and Moderna use lipid nano carriers for in the mRNA vaccine formulation. So what kind of impacts or what kind of questions result in from this beautiful technology? It's first of all environmental, health and safety concerns as a result of engineered nanoparticles, because we do not know all so far all functions and all properties once we go down to nanoscale. And there is a very strong requirement for to study nanoparticle fate from cradle to grave, so including biocompatibility, biodistribution, and then further nanoparticles and how they interact with environment. So ultimate goals for everyone who is working in nanotechnology can be formulated as Richard Smalley, the Nobel Prizer, who was awarded for discovery of buckyballs. He actually formulated it in 2011, and it's still very important. So nanotechnology goals to enhance energy technologies, water, environment, and diseases. And also, as scientists, we always advocate for education, democracy, and here's like main threats identified by the United Nations. And you can see time goes, but we still have these very important questions, concerns to address. Nowadays is infectious diseases is very important, a very important part of our life. So since I'm working mostly in nanobio hybrids, I would generally say how they work. So we look at nanobio hybrid upon external stimuli applications like light or X-ray or magnetic field. These particles can produce induced electrons or ions or voltage or light of certain wavelength or heat. It could be also mechanical torque or mechanochemical process. And all these can be utilized for signal transduction through a biological matter for drug delivery, theranostics, sensors, and nowadays is very important brain technologies. But you will be surprised, similar mechanisms in very often very similar nanobiostructures can be applied for green technologies like artificial photosynthesis or new source of energy and computing and etc. So in our group, we are dealing with nanobio interfaces at different level of complexity. So first row is molecule particle, for example, DNA combined with magnetic nanoparticle. Here is very cool image. It's a adenovirus decorated with magnetic nanoparticles. So we can go further to cell particle combination with just a different row of like heroes, for example, I pick up. It's neurons with nanophosphors inside of animal brain. So, and also further, it could be combined with, we can absorb particles or we can introduce them to whole animal. This one, for example, this image in biomaterials, it's then emergence of liver cancer, shine or just lightened by upconversion nanoparticles, and surgeon can use just light to cut with high precision, cut the tumor. And now I'm going to show you one example after this introduction of our own work. We recently started working toward technologies for brain control, brain studies, and also brain disorders. So in this, I would like to show you, it's a traditional method approved by FDA, deep brain stimulation. Then electrode is inserted directly to human brain. And also there is a pacemaker-like device, it's a controller. Some patients with no way of treatment, but are managed by this approach. So first of all, most important is Parkinson's disease. And last years, nanotechnology came to aid this field, and nanomaterials were proposed to be used as mediators to respond to external stimuli like light, ultrasound, or magnetic fields. And there are a lot of bottlenecks, issues in this field. However, there is a step that nano aids neural modulation and DBS. Like around 15 years ago, more than 15 years ago, it's two key publications reported upon optogenetics. This is technology that utilizes light to control genetically modified neurons. So basically, this is an example. If then usual ionic channels in brain can be infected by viral virus-based vector, and this neuron is getting light sensitive, responding to certain wavelengths. So one part is genetic modification, and another part is optical guides, and they still, or optical fibers, they still should be inserted inside of the brain. And we asked ourselves, can we just develop something different, really wireless? So since we work in our big part of Argonne is advanced photon source and X-rays, we ask ourselves, what if X-rays such as X-rays from usual dentistry office machine can be used for wireless optogenetic? So here is it's just a cartoon. It's not real. It's not biological tissue. So we developed system. Then we used opsin or ionic channel responsive to near infrared light, and also we developed nanoparticles which can absorb energy of X-ray and emit, transform it to energy of photons. We tune them exactly to overlay with action spectral window of the opsin, so 612 nanometers. And we demonstrated, so we synthesized, we developed these particles. These are like rice-shaped or spindle-shaped nanoparticles. So we totally characterized them. I don't have time to show everything. And we demonstrated in long experiment, it's several weeks, a few months, using group of animals that brain, animal brain responds to train or just sequence of X-ray pulses with latency about 100 milliseconds, which is very good result. So means it's a very quick response of brain to X-ray stimuli. And also we demonstrate that specific markers overexpressed as a result of then brains, actually neurons, are induced for extended time. So it's important to show, since I understand there are a lot of standard methods to evaluate stability, biocompatibility, and chemical inertness of nanoparticles in vivo. I'm not mentioning them, so I want to show you X-ray fluorescence, which is a super resolution and super sensitive method, synchrotron method, with super special resolution down to dozens of nanometers. So each pixel is only, in this case, 40 nanometers. So we demonstrated after a few months of residing particles in animal brain, they are still stable. They still have a very strong signal of even trace elements we used for doping these particles. And also they are located very close to neurons. So this is very important. This is an example of collaboration between multiple universities. It's University of Maryland and also University of Pittsburgh. Neurobiologists came to us, and nanotechnology, and also synchrotron and X-ray sciences. So I got a few answer of questions about how nanotechnology is affordable for third world countries, and I would like to emphasize. So even in at the level of CNM, Center for Nanoscale Materials, you see we have a worldwide distribution of users from Africa, from Australia, from South America. We have a lot of people even from Western Europe. And if we are talking more about how many countries pursue nanotechnology, I would say this is going to be really a big list of countries. So, and I would like to show this image of my colleagues, and I would say nano really unites us. So together we can. Thank you.
Thank you for that, Elena. Let's go back and introduce Jinwoo Kim here for a second. Jinwoo Kim, full disclosure here, is serving as the vice president of publications for the IEEE Nanotechnology Council. He is also a professor of biological engineering, biomedical engineering, biological engineering, biomedical engineering, and nanoscience and engineering at the University of Arkansas, and an adjunct professor of electrical engineering at Pohang University of Science and Technology. Obviously, he's got a lot of very distinguished aspects to him, so I'll just touch on a couple larger ones here. Obviously, again, he's serving as co-editor-in-chief of the IEEE Open Journal of Nanotechnology, which finally got its first big spotlight in the Scopus Index over the past quarter. So everybody in the NTC is very excited about that because it's a very new journal, open access. So we're hoping to engage in research through that medium whenever possible. And he, again, is just very engaged in the many of the conferences that are held year-round, including the NMDC, which is Nanotechnology Medical Device Conference, which this year will be held in December. So I'll stop rambling on and hand it over to Dr. Kim.
Okay.
Thank you. Your kind introduction, Tyler. Let me share. Can you guys see my screen?
Yes.
Yes.
Okay. Hi, everyone. I'm Jinwoo Kim, and thank you for inviting me for this exciting event. Today, I will discuss key items to consider when designing and synthesizing nanoscale materials for medical applications. Due to the time limit, I may not go over details of technologies, and most of my slides does not contain many technical details, but I'd have to further discuss during Q&A session or personal email exchanges. And certainly, Dr. Roshkova's wonderful introduction about nanoparticles and their interface and their applications would, I believe, certainly help understanding my talk. Okay, during the last 2 decades, various nanoscale materials, including carbon nanotubes, Q-dots, various inorganic and organic nanoparticles, and so on, showed immense promise for more effective treatment and management of many diseases, particularly like cancers. And a new term, nanotheranostics, was evolved in about 10 years ago. Nanotheranostics mean nanotechnology-based therapeutics and diagnostics, nano from nanotechnology, and thera from theranostics, and gnostics from diagnostics. Do theranostics and diagnostics together. As illustrated in the cartoon, nanoscale materials usually decorated with functional materials to target and treat diseases. And then these materials include antibodies, drugs, and as such. And when injecting them to animals, they guide the detection and treatment of diseases. I'm going to use our previous studies as examples. And the first, I have to say these studies were done in collaboration with medical scientists, particularly Dr. Vladimir Jarov at University of Arkansas for Medical Sciences. First, the carbon nanotube responds to near-infrared light, showing promise as non-invasive contrast agents. This is because biological tissues are relatively transparent to near-infrared light. In other words, normal tissue not absorbing near-infrared light, so no or minimal damage to healthy tissues. But our carbon nanotubes have relatively low responsiveness to near-infrared light, and also there are issues related to their toxicity. So to meet this challenge, we developed the gold-plated carbon nanotube. We call that as golden nanotubes by fabricating a thin layer of gold around the carbon nanotube. This new hybrid material showed drastically increased their infrared absorbance, actually around 100-fold increase compared to carbon nanotube, and did not show any apparent toxicity. In animal studies, we imaged and mapped lymphatics for the first time by detecting thermal and acoustic signals from golden nanotubes targeted lymphatics. And also by increasing the intensity of near-infrared light, we demonstrated eliminating damaged cells, and this showed potential for non-invasively treating diseases. This study serves as, I believe, an example how to improve theranostic materials by uniquely merging different materials. In our case, two different types of materials, gold and the carbon nanotube. Next, by combining golden nanotube and the magnetic particles, we showed an in vivo real-time theranostic platform that can capture through magnetic enrichment and detect by near-infrared the responsiveness of a golden nanotube of rare circulating tumor cells. Metastasis caused by circulating tumor cells accounts for over 90% of cancer death. But early detection of these circulating tumor cells is very hard, and we commonly say it is like searching a needle in haystack. Metastasis is really challenging, and circulating tumor cells are extremely rare events, and very easily obscured by overwhelming background materials in whole blood. And also our human has a large volume of blood, around 5 liters. And imagine finding a few cells in 5 liters of liquid. It's a really, really hard task. So the ex vivo analysis currently existing, the sensitivity is excellent, but it has certainly limit. So our work provide a unique way for early detection and treatment of metastatic cancers and infectious diseases. And I think this study is another example of showing how combining materials with different functions could lead to clever ways of overcoming current challenges and making a breakthrough. With the lessons from these studies, we started developing strategies and methodologies to design and assemble multifunctional nanoagents by controllably combining different nanomaterials and producing hybrid nanoagents with customized or tailored size, shape, and functionality. So to design the effective multifunctional nanotheranostics agents, we should consider not only materials themselves, whether they are suitable for targeted application, but also biological challenges that externally administered nanoparticles in a complex biological system when they injected into. As shown in the left box, recent advances in nanotechnology have yielded various nanoscale materials with different shapes, sizes, and compositions, and their functions or properties depend on their composition, size, shape, surface chemistry, etc. Because of this, proper selection of nanoparticles is one important key to fabricate the nanoagents for a specific target application. And also biological challenges must be considered for the design and development of multifunctional agents. And as shown in the right box, the behavior and the fate in very complex and heterogeneous biological systems should be considered. For example, abundant proteins in biological systems will interact with nanoparticles when injected and could change or influence their properties and functions. And also what the immune responses to nanoparticles should be considered, and how long they will stay in biological systems, their biodistribution, as well as their toxicity should be considered. But based upon the studies so far, it was found that these biological challenges, most of them, if not all, could be overcome by properly controlling their size, shape, and the surface chemistry, in other words, their property and the functions. So in designing multifunctional nanoagents, the key parameters to consider is size, shape, composition, and the surface chemistry. These determine not only their fate in biological system, but also their intended functions as a medical agent. So developing effective, efficient strategies to design and assemble functional nanoagents with the designed shapes, sizes, and the compositions is a very important step. There has been significant progress were made during last 2 decades, but so-called controlled synthesis or construction of such functional nanostructures with a specific or designed size, shape, composition is still very challenging. So in our group, to meet the challenge, we are developing so-called nano toolbox technology for programmable or more controllable assembly of such multifunctional nanoagents. Left-hand side, general cover illustrates our core technology to control number and the geometric locations of distinct functional ligands. These functional ligands connect particles together. And in our study, we are using DNA as functional ligands. And these functional ligands on a particle with the control over number and the geometric location. And we try to achieve programmability, which is a key for programmable assembly of particles in controlled matters. We first demonstrated this concept with the colloidal gold nanoparticle, and we achieved with the so-called one step at a time process. Briefly, by attaching one DNA at a time to the particle, which we achieved the site-specific binding of DNA to nanoparticle surfaces in a stoichiometric manner, and fabricated the building blocks with the defined number and type of DNA linkers at designed locations and orientations. And then we generalized this technique to other types of nanoparticles. The work is still in progress, but we had quite a bit of progress up to now, and we generate so-called nano toolbox with the building blocks of various nanoparticles are generated. And with this nano toolbox, our approach enables DNA-guided programmable self-assemblies of multiple nanoparticles into multifunctional nanostructures with customized or tailored size, shape, and functionality. And I believe it can drive innovations in highly sensitive and multifaceted and multimodal with various medical imaging or therapeutic instruments. They can advance medicine, nanomedicines for optimized treatment and diagnosis. One example would be given various possible combinations of nanoparticles in nanostructures, multiple tasks and functionalities can be provided in sequence or in parallel. So this could allow harnessing complementary ability of different imaging and therapeutic modalities like MRI, CT, X-ray. Each has unique advantage, but also what they have disadvantages. So all simultaneously doing so, we could achieve a more comprehensive, accurate, and reliable treatment and detection of diseases. So in summary, the merger of desirable functionalities of various nanoparticles in controlled matter and producing hybrid multifunctional nanomaterials with customized or tailored size, shape, functionality. This carry enormous potential to advance the field of medicine. To realize this potential, but we should take a holistic approach that consider not only hurdles regarding chemistry, control of nanoparticle assembly, but also still incomplete, but growing understanding of biology and the interactions between nanomaterials and biological components in complex biological systems. I believe this holistic approach and the advancement of technology and science in this field will lead us to designing and developing customizable, tailored, designed, multifunctional agents for successfully develop and translate to critical stage. Thank you.
So thank you both very much, Professor Kim and Dr. Rosenkova. I'm very excited. Actually, I work in related field, and actually, as I said, we do a little bit about biological research, and some of the topics you both mentioned are really exciting to me personally. So thank you both very much. So now we're open for questions, and I know we have a few questions already in chat. Should we start with that, Chris, or should we? How do you want to manage this?
Oh, we can start from the question in the chat. So the first question which is there, which is fascinating because it's a medical doctor, is about using the nanomaterials to help the bone growth.
Yeah.
So I saw Elena kind of answer that question, but if we can build on that and maybe everybody can chip in, will be great.
Yeah, I think that would be helpful if Elena can, speak out about it, like instead commenting by chat.
Well, let's read out the question for the audience who can't see the chat here. So, could nanotech lead to therapeutics for broken bones, glue or mesh around the break, or a sort of inner cast for the broken internal structures?
So can I?
Yes, please.
It's a great question. And I believe there is it's a quite vigorous development in materials in biomaterials, and like scaffolds. Actually, they can be composed of inorganic materials. They can be there are plenty diversity of scaffolds, including biological polymers and synthetic polymers combined with inorganic particles. And such scaffolds, they sometimes in the shape can be fabricated in different shapes, and they can be uploaded with drugs, anti-inflammation drugs, some factors which enhance bone tissue regeneration, and also they can be uploaded with stem cells. So I think it's a big area to explore, and I recently explored it myself.
I totally agree with Elena. My group in part involved in that too. But there is a lot of study have been done, and particularly bone regeneration. That's what really one key challenge in medicine. And our approach is what somewhat relate to the presentation I made. The key in tissue regeneration is how cells better attach and grow similar or same, supposed to be same as native cells, right? So that can be, relate to the material we apply. Elena said the scaffold. So what types of material we are using, and also on the surface or internally, what the overall structure there, right? So that the cell attach better and the differentiate to intended the type of cells. So there had been a lot of study done, and also what I saw clinical study of they call that as patches. That's actually scaffold, and then what utilizing that for regenerating the bones.
Did you have a comment, Chris?
Dr. Kamps, you first.
Oh, no. So what do you mean by a scaffold? Aren't we talking about very small materials? So how do you turn a nanomaterial into a scaffold?
So that is what the scaffold is overall is what the not small material, right? So bone damage there, then what there is relatively large the membrane type gotta be applied, but nanomaterial will be part of the material to make such membrane over scaffold. So nanomaterial is part of the material, but what the scale of the scaffold is not nanoscale. But internally, nanomaterial is there. Nanomaterial contribute the function as well as what the surface chemistry and chemical and physical properties there, so that facilitate cell growth.
Wonderful.
So I have a quick question. Now, one of the frequent problems that we face in the clinic, especially in the bone, is the bone infection, right? And quite often what happens, some other guests, something called osteomyelitis, and now we have this kind of horrible choice that, yes, I have to take the hardware out from the patients, and I have to put the new hardware, which most likely will get infected, and all the process of healing takes forever. I guess my question is there advantage? So one of the treatment is to put the local antibiotics, which I understand we can load the nanotech devices the same way. But my question is slightly different. Is there any inherited resistance of the nanomaterials to make these infections less likely? Because every single time I'm going to put the screw in the patient who has osteomyelitis, which was infected first, I'm going to have the same problem. And I can put all the antibiotics I want, but I still don't change the overall problem.
Can I answer?
Yeah, go ahead, Elena.
Of course, if we tell you today what you should add instead of antibiotics, it's not going to work like that immediately. So it's a process of development. But many of nanoparticles, especially metallic nanoparticles like silver, they demonstrate very strong antimicrobial properties. So if they are combined with mesh or with scaffold, whatever you call it, so they can serve as a local center to kill bacteria. And obviously it should extend work of your implanted implanted device.
I see.
Maybe.
Yeah,
I was just going to ask because this was something that I'm researching, obviously, as the more theoretical aspect of these things. Is there the potentiality for nanotechnology to serve as a sort of a compoundless anesthesia, or a technology that is able to pinpoint a very specific part of the body and generate an anesthetic-like effect so that we're not having to use these drugs, these opioids in the ER when we're going in and treating patients? And if so, what's the timeline you guys see for that rolling out in mass? Maybe Jin first and then Elena?
Well, really good question. And that's another important area to tackle, right? So certainly it's possible. That is what, but it really depends upon how much we understand the biological system, right? So to apply nanotechnology or what the nanoparticles to do what the such function there, then what the location we target, for example, what the nervous system there, and go there and the target and bind and block the signal passage. That's certainly possible there. But what, as far as I know, I haven't seen any report yet, about the applications of nanoparticles or nanomaterials, for such application there. But certainly, it's really, feasible area, and but I don't know how long it's going to take. That will depends upon how many people in this research community get involved, and that work certainly needs to be done in collaboration between material people and the medical area as well as the biology. So, Elena, if you have any, thing to add, please.
So, I know there are some bulk of data on biodistribution and clearance of nanoparticles from different organs, even from brain. I know. One I have in mind. And for every approach, for every single case, with biodistribution and retention of particles in vivo should be studied before it goes toward preclinical. But it's a long story. But I know for many of nanoparticles is there is available data on clearance of particles and retention of particles inside of body. And it should be reasonable time, yes, and particles can surf and then can be from
Are there any reports of
inflammation.
Are there any reports of nephrotoxicity? Because you'd think that as they're being cleared, these particles could be retained in the kidney.
I am not aware of this. I did not study this field much about kidney toxicity.
I think the toxicity is what always the issue until we find everything with the long-term study, right? So including our study, we do define the period of time and whether it causes problem to normal cells or not. But that is merely 1 month or 3 months. So during that period of time, we say we don't see any apparent toxicity, but we never know what is the long-term impact there. So definitely we need the long-term studies. But one important thing I believe is the best thing I believe is particle get into our body system, do the work, and get out of the body, not sequestered in liver or kidney, right? So in order to do it, that size, shape, the surface chemistry is very important. So for example, studies tell that particles less than, around the 5 nanometer, some say around 10 nanometer, smaller one comes out of the body. So size is very important too. So how we can design the structure so that we can make those particles cleared out of body. I believe clearing from the body is the best option.
Can you program them to self-destroy?
Self-destroy is, I think it's possible. One example is our approach in the infrared light, right? So one study we did was the gold particle and targeting, and then increasing the intensity of light, actually gold particle explode, right? So that would be one way, but there's a downside there too. To explode the high intensity of light there, then it hits up to over 100 degrees. So then it could damage cells, right? So there is different ways. So our approach is rather than using large particle themselves, using smaller particle like less than 5 nanometer, and then through our technology and making overall shape, done the work. Then since we are using biologically degradable material like DNA, so eventually structure disassembled, you need particle less than 5 nanometer, so it could get out of the body. So that could be one way.
Wonderful, thank you.
May I just also add a little bit? I think very often scientists working in nanotechnology have to do something to extend time of particles in bloodstream because in overall body likes to get rid of particles immediately. So for that,
that's right.
I agree with Dr. Kim. Surface should be modified with specific brushes to hide particle and let particle to do job inside of body before this particle is cleared from the body. Also for inorganic nanoparticles, for example, one of most known, it's for general audience, it's magnetic nanoparticles, iron oxide. So iron oxide can degrade inside of body using natural body systems, and also I know one of FDA approved iron oxide initially developed as MRI enhancement. Nowadays is used as a source for iron deficiency. So nanoparticles can be used because they allow for slow release of iron and support the body if there is not enough iron inside.
Mhm. Wonderful.
Can I ask very sort of naive question? Why do you need to clear nanoparticles in the first place, right? Like, so this is maybe my very simple doctor-like imagination. Like most of the time when I hear about the nanoparticles, they usually drug delivery device. When the stuff is delivered, well, they can float, is a debris, right? So, you know, maybe this is me a little bit being, you know, very simplistic. And remember, this also goes to other doctors. So we sort of like wonder, you know, how much of this, you know, how much of this size matter. I can also tell you my personal story when somebody approached me at Penn and said, you know, we have this new COVID vaccine, which is based on the nanotechnology. I was like, well, I think I opt out from that one. Okay, because I was like, there's absolutely no idea what that's convey. So I think my question is like, what do we know about danger of having nanoparticles inside you for like forever?
That's a good question, and maybe not, right? But as scientist, my you know my thinking towards that is related to toxicity. Until it is proven being non-toxic, we should consider that as toxic. So that's why you know, we need a long-term study how it's going to influence into the body, right? So for the vaccine case, the liposome they are using, you know, some of them they are using biological material, right? Biological lipid and such. Since it is you know, biological material, in other words, but it's part of our body system, so their influence on human body would be quite minimal. But inorganic or organic particles, that's totally foreign material there, right? So how it's going to influence the body for long term? Usually, such particle at the end up to liver or spleen. Those are what the locations those foreign matters accumulates. So we should study long term, you know, the influence. So, but at the end, who knows what they don't do anything or what some good thing for our body, never know. But until it being proved, we got to consider that as toxic.
Thank you.
Do you have a question to present to Dr. Zhang, or should we go off of the list from the pre-selected questions from the registration?
I did get a question from one of my colleagues on the WhatsApp. So if I understand this correctly, there are natural nanotechnology devices like ferritin inside us, correct?
Yes.
And how many other sort of natural occurring nanotechnology thing happens? Because like literally, I'm reading somebody who said, I thought this is all artificial, and now we seem to be the natural person.
Nanostructures, right? So in natural systems, nanoscale nanoparticles could be good and could be like bad example of biomineralization in brain during Alzheimer's disease development because of sticky peptides or plaques formed in brain. They also help deposition of ions and formation, biomineralization of, for example, iron oxide nanoparticles. This is quite known. So as for good examples of nanoparticles formation, my favorite is bacteria, some bacteria that can switch breathing or respiratory cycle to anaerobic and use some salts as a source or as precursors for nanoparticles formation. So bacteria needs to get rid of excess of electrons and sends them back outside of bacteria, and nanoparticles form around with bacteria. So it's like sinking of electrons. It's a very smart system. So ferritin is a natural nano cage serving for storage of iron in not biologically toxic form inside of body. So I'm not mentioning virus. Every virus is a very well designed nano system with very precise size distribution. Every virus is like same like another one.
Now, one additional example I like to add to is bacteria case. There's a group of species, so-called magnetobacteria. So they respond to the magnetic field and as such. And if you look at inside such microorganism, they synthesize a small, well-defined magnetic particle inside the body. They're system. So that would be another example, naturally synthesized nanoparticles.
So I would like to add more. Maybe it's not a kind of advertisement of our work, but it's just excitement about our work. So a couple years ago, we reported on synthesis of gold nanoparticles in hypoxic brain. So we used isolated living brain and organized like artificial hypoxia conditions, not enough oxygen were provided. And since we knew biochemistry of this process, we actually add precursors of gold and brain. It was animal brain synthesized very well size distribution of gold nanoparticles, and we actually confirm it using advanced SAX-VAX methods, synchrotron methods showing high crystallinity nanoparticles in brain. So it could be applied for example, for imaging, for tests, or hypoxic organs for organ transplantation, something like that. We also patented this method.
I can tell you that a lot of my colleagues do not realize that. So what you guys talking about is a little bit something between fiction for them. For you, it's science.
Maybe I can add a quick example from the optical perspective, since I do spectroscopy. Opal, there's a lot of so-called photonic crystal structures. They are very periodical structures on the nanoscale. Of course, they're sort of larger on the hundreds of nanometer scale. But they could be made smaller for other purposes. So these are natural, a lot of natural nanomaterials, put that way.
Yeah.
Okay, let's just continue with the questions. Tyler, are you monitoring the questions? I'm looking.
Yeah. So I'm looking at the list that I have pulled up here, and I mean, we can probably mix this one together. So a dual part, is using nanotech to enhance humans problematic? And, in tangent to that, why should we be talking about ethics of nanotechnology if this is just a smaller scale of engineering more generally? Maybe Elena first and then Jinwoo.
So actually, I also I read these questions, and as I mentioned it during my introduction part of my talk. So we have to care about safety and ethics and all kind of issues. Then we are dealing with nanoscale materials because their properties are not completely studied yet, and they can be very different from both from bulk material and from same composition, but bigger particles. So that's why every time all these concerns should be addressed.
Yeah, I agree with Elena. I mean, that Elena well put, and particularly the safety-related issues, there is still, a lot of questions there. And applying, the non-proven material to human body itself is what I believe not really ethical. So those safety and related ethical issues should be studied and also discussed among the community as well as the users.
Yeah.
I be a little bit advocate diaboli over here. Like, look what happened with, we always talk about the ethics being very, very cautious, right? But then we have a COVID-19 pandemic and this incredible train of 20 different vaccines, at least 3 of them based on the technology which was never based, we never tried before. And then we pretty much roll it out. And I think it's very interesting how you hear the message. Being critical care doctor, it was fascinating from one side here that, yes, this is a safe vaccine, and then pretty much sign the waiver that we have absolutely no clue what this does to you. But one of the things happened is that turns out that was a pretty good actual trade-off at the end of the day. And when I'm sort of hearing let's wait for the more safety, I'm like, when it's enough safety, right? And maybe sometimes, there may be more dangerous things around this. And maybe, the beauty of the nanotechnology is that, benefit risk ratio over here is better, like with the COVID vaccine, right?
Well, if I can just caveat that just really quick, because there is concerns specifically because the mRNA pathway that they're using is targeting what we consider in the medical field the short-term memory of the immune system. So the concern is that by targeting the short-term immune system, it doesn't allow the body to build up a natural immunity, or it requires exposure to gain natural immunity that can't be attained through the mRNA exposure. So there is this concern that, yes, we are vaccinating ourselves for the short term, but then we are necessarily needing to get the quote unquote booster shots, or the I guess we should say reminder shots is probably the better semantic term for, because we're just refreshing the immune system's memory on how to fight this virus and the dangers that it might pose to the system. Sorry, didn't want to steal anyone's thunder. I just given the perspective from the medical aspect of things here.
One of my questions would be, if I come to Jean Wu, or Elena, and I say, okay, I would like to take the particle which deliver oxygen to brain, because one of the problems I deal with is the TBI in my patients. I know is there a regulatory process which even covers that? Because we have one interview with somebody who invented AI in the United States, and he spent 10 years inventing with FDA how to actually get the AI approved. So I'm thinking, okay, paperwork already is 8,000 pages for the regular compounds. For something like that is going to be 16,000 of the free creation by somebody, or we should just do it.
Yeah, that same apply to the application of any material to human. So that preclinical and the clinical trials, and there's 3 stages there, right? So it's a lengthy and tedious process there. So even we, show real fantastic and magical, the formula, but to apply to human, we gotta go through the regulatory process. So that's the thing. That's a really. I don't think it's necessarily bottleneck. It is important, right? It gotta be tested thoroughly before applying human, but it's in a sense, the bottleneck to the timely translation of technology to the clinical application. Yeah.
Can I segue this really quick to the last poll question? Because I know Elena talked about this a little bit in her presentation, but how expensive is it really to perform research in nanotechnology? How can we ensure that developing countries can compete in this technology when a lot of them still don't have sufficient broadband internet access? Is this just yet another technology that is going to divide the rich from the poor, sort of thing? So Elena, if you would please first, and then Jim, if you have any extra comments.
Yes, science is expensive. So because of science is expensive, and we all do hope that we are using taxpayers' money, not just for fun, but we are using for future to help to increase or improve like lives of national wise and actually globally. Today, after I got this question, I did talk to our management and ask how much we spent per year. Our division, it's a Center for Nanoscale Materials, working by ourselves and for users. So it's around $28 million per year. So this is a very big money. And however, I would say I visited institutions and nano centers in India and in other countries, and they are doing just great. They are doing great, and even for smaller money. So I would say, yes, it's expensive. However, there are great science, great publications, and developments are coming from around the world, from Asia, from South America, from Western Europe. And I believe, of course, we have to continue.
Yeah, I agree. I mean, that is what indeed science is expensive, and the availability of funding will determine not only the money to carry out the research in individual labs, and also what core the instruments, like what the fantastic the equipment in Argonne National Laboratory. Without it, we cannot do the really sophisticated work. Those infrastructures should be supported by government and or what the other big company at least. But in developing country, certainly there is the limitation there, and also what some developing country, the communication with globally, there is a certain limitation there, but it's getting better and better. So, such difficulty in communication and collaborating with people in a bit more advanced country. I mean, that is what another thing should happen. But it improving, but certainly there is a limitation there. But I agree with Elena. And I've seen a lot of work, good work published from countries in South America and some Eastern Asia and as such. So it's a promising, but there's certainly a lot of improvement needs to be done.
Yes, we are not mentioning that we have scientists here in US. We have scientists from around the world working in US or in some foreign labs and universities, and there is a totally diversity, inclusion, and equity opportunities for everyone, for talent. So for talent and for passion about science. Passion.
Well, I think that's a very nice ending, very optimistic, very against the crowds, very juxtaposed to the current time when everybody seems to be somehow irritated at each other. So I think Elena very nicely, wrap it up. how the nanotechnology not only give us hope in the medical field, but also brings this positive humanistic value. Thank you very much for that. I would like to thank you everybody for having this fantastic session. I got a lot of questions on the WhatsApp from my colleagues and friends. We have a wonderful speakers. Probably people will get back to you since you guys are on our YouTube with the more questions. And please enjoy your evening today. Have a good one. Thank you.
Thank you.
Appreciate